I'm presenting a mini oral presentation in the translational research session on the critical role of eosinophils during response to immune checkpoint blockade. So we know that only a minority of patients with metastatic triple-negative breast cancer responds to immune checkpoint blockade. So we need a better understanding of the mechanism of response. And while most studies focus on the tumors and tumor microenvironments, we also know that systemic immune cells play an important role. But the role of myeloid cells particularly is poorly understood. So to investigate the role of circulating immune cells in response to a checkpoint blockade in these patients, we investigated peripheral immune cells of patients treated in the TONIC trial by high-dimensional flow cytometry. And these are patients with metastatic triple-negative breast cancer treated with anti-PD-1, preceded by a 2-week induction treatment. And we took biopsies and fresh blood on several time points. Because we performed the analysis fresh, we were able to study both lymphoid and myeloid cells. Parallel to this, we also performed mechanistic studies in mouse models. So what we found was that there was a systemic increase in eosinophils in patients responding to anti-PD-1 on treatment, but not in patients not responding to checkpoint blockade. And we also saw that in paired biopsies of these patients that there was an increase in expression of eosinophil-related genes in responders, but not in non-responders. To investigate whether eosinophils play a causal role in response to checkpoint blockade, we used 2 mouse models developed in the Gade and the Visser lab for both early stage and metastatic disease. And in these mouse models, we observed that there were high levels of eosinophils in the blood of mice that were treated with checkpoint blockade. And generally, these models respond very well to dual checkpoint blockade combined with chemotherapy. But upon depletion of eosinophils by anti-cyclic F, this effect was completely abrogated, indicating that eosinophils really play a crucial role in response to checkpoint blockade. And finally, we validated our findings in several other clinical cohorts. And we observed that there was an increase in systemic eosinophils in patients responding with metastatic bladder cancer and metastatic non-small cell lung cancer. And also in patients with early stage mismatch repair-proficient colon cancer treated with neoadjuvant checkpoint blockade. We observed that there was a systemic increase in eosinophils only in responders. And also in the tumors of these patients, we observed that there was increased expression of eosinophil-related genes upon treatment in responders only. And in patients with early-stage mismatch repair-deficient colon cancer and in patients with mismatch repair-deficient metastatic cancers, we did not observe a systemic increase in eosinophils, indicating that it's context-dependent. So altogether, these findings demonstrate that eosinophils play a crucial role in response to immune checkpoint blockade, and that they can potentially be engaged in future combination treatments to enhance response to checkpoint blockade.